You see, even the terms we need to be able to say.
Yeah, I think I'm good. Yeah.
Perfect.
So one thing I can promise that in the end of this talk you'll know all those words. And so really my goal is to make it accessible to everybody. It doesn't matter what's the background. So I don't mind ending my talk in the middle and just explain. So I really want people to understand what we are talking about. And the topic that I picked is on revisiting, thinking back on predisposition genes in cancer. And as you'll see, cancer will be only a case study. So it's beyond a cancer story. So let me just get going. So basically, at least the first part of my talk will be on cancer, just to understand what we're talking about, on genetics, because most of this talk will be on genetics, and on evolution. And actually, I want to share with you our thinking on evolution as being a driving force to understand what's going on. So it's a little bit, I'll push this point a little bit further, and specifically, I'll talk about cancer in terms of evolution, not in terms of disease and survival, and sorry for the epidemiologist in the crowd, I mean, it will be a bit different. Then I'll introduce a tool or a concept or platform that we have developed to get to understand the selection, evolution selection within cancer. Then I'll move to PWAS, which is our method to understand human population. And the idea is really to move from cancer sample to population, and then maybe to private variation, a little bit connected to the privacy that we heard. I mean, these very unique variants. And then something about family, common, rare, and what will be next. Okay, so let's start going. So I like this statement, which is almost 50 years ago, saying nothing in biology makes sense except in the light of evolution. It was mentioned by some famous, it was in the church, a very famous guy in the church. He believed in evolution. And I wanted to show you that it's a good thinking, mode of thinking, to think about cancer as an evolutionary process. What I mean by that, let's say, just for that this is one cell, normal cell, healthy cell, and at one point there are mutations, and each mutation takes its own roots. And at a certain point, there is another mutation that gives this color, and then another one, and another one. And basically, this heterogeneity in the cancer is a dynamic process that started from nothing, from the first event. So you can formalize it in a better way just by showing the same picture, less colorful. And you can see those events. Let's say this is the primary tumor. And indeed, if you just check the tumor, you will see many colors. Colors means different cell types, different states. But let's say this guy, this cell, the green one, became to be, gain some benefits in order to become metastatic. So now the same cell that was randomly mutated and so on became to be with a better fitness. This is why I'm using this evolutionary as a framework. Okay, I think you got it, and that's important. So today, we are dealing with a lot of what happened to the genome, which is part of the cancer, is the instability in the genome, of course. And what I want to mention that today, just for simplicity, I will only talk about mutations. By the way, they are not the most important part. Deletion, amplification, I mean, more parts of a chromosome, less part of, I mean, really changing in the overall structure of your genome is important. But today, I'll talk about mutations, okay, which is single event. Okay, so going back to evolution, so this is just to show you that I can use the same terms from evolution, which is selection pressure. For example, there is a competition on resources. Some drug pushes the system a little bit like bacteria that we are putting antibiotics and then we shift the balance of the ecosystem. This is why I want to use those terms. It is a dynamic process, so mutation rates become to be very important. I mean, how fast it's all happening. And of course, I would even dare to push the argument and say that cancer progression is a genetic alteration and fitness, the combination between changes and how good it is for the cancer itself. And this is just to show the same picture that I showed before. In a more cellular view, you can see that at some point, this red one become dominant. But it was at a later stage, let's say, after treatment, some drug treatment, okay? So I think you have it. So I wanted to take you a few years back to the COVID time, just to show you that it's not unique to cancer, what I'm telling you. It's unique to any ecosystem and biological dynamics system, which is everything that we study throughout my life. So if you look at the evolution of SARS-CoV-2, you can see this is the first year in Europe. What you see is here we are in 2020. There is one variant, and immediately, a month later, another variant and another variant. And here there was this bad guy that came back. But you see that they are competing each other. So at any certain time, you have heterogeneity, exactly as I showed you for the cancer story. And actually, this picture that is crazy, it's like 2 years within the COVID. And you can see the rise and fall of the Reich, you know, the rise and fall of this variant. And here, the Omicron that is still around came back, but you can see the dynamics in front of our eyes just by looking at this colored variant that I showed you for the cancer. So this is the analogy that I'm trying to share with you. And at some point, this is the same view as this, just showing that it started here and it's immediately become branched and colored. So I think this analogy is really important. And the last formalization that I want to use is the term that people in the cancer research are using, which is drivers and passengers. Drivers means the genes that indeed, if they are affected, they make progress of the cancer. And the passenger just hitchhikers. I mean, they just jump over, and apparently in a simplistic way, I mean, of course, they do not contribute anything to the cancer, let's say. But importantly, in cancer sample, if I take biopsy and I check, there are only few drivers, maybe one, maybe two, but hundreds of passengers. And one of the questions that we ask ourselves as a scientist, can we separate them? Can we know who is who? Okay, so this is, yeah, please, I mean, feel free to interrupt.
So I should think of it as basically simultaneously a bunch of mutations happen at once, and you're trying to understand which one is conferring the
I even
advantage.
dare to say, let's say, which is not true, but just for simplicity, and let's say mutations are coming like a shotgun, like everywhere. Mm-hmm. So most of the mutation will be, okay, nothing happened, but from time to time, and this is the driver, if you hit a gene that is really responsible, and I'll give an example in a minute, responsible, then the entire fate of the cancer will be changed. Okay, so basically there are a lot of those noise. Let's say it's not noise, but the numbers are important. So let me tell you what's the fitness is all about. I mentioned fitness, but fitness for what? So fitness, by definition, is a way to improve your children's success or whatever in society. But cancer, I'm now putting myself in the head of the cancer. Cancer likes to do, for example, inducing angiogenesis, which means attract vessels, blood vessels. Therefore, the cancer will get more oxygen and can thrive, or for example, resist apoptosis. Apoptosis is a controlled cell death. So no cell death. So this is a benefit for the cancer, let's say. So all those, we call them hallmarks. Those are the 10 cancer hallmarks. Sometimes you can have more. But for example, a very important one that now we are researching, but it won't be for this talk, unless you would ask for another one, but it's okay. It's invading the immune destruction. So cancer, the immune system is very potent in doing it. We know it because of the immunotherapy that is so successful in some cases. But the ability of the cancer to invade this gives a huge advantage. So those are the fitness map. And now I want to move into the molecular map, the gene map. What do we see? This is an illustration of cancer, of lung cancer, breast cancer, and so on and so forth. And if you just look at it, you see that some names are repeating, but each of them has its own set of genes that are, now I'm talking about the drivers, as I formulated before. So for example, if you look, you don't have to be very smart to see that P53, for example, appears in quite a lot of those, right? Of those different, and it's not by mistake, it's real. And let me show you what P53 is doing. I'll use it as an example for very strong driver, but only to run my arguments, okay? So it's relevant to many others. So P53 is a very well-known cancer gene. The reason being is because it's a sensor to a lot of things that happen under stress. For example, hypoxia means no oxygen. Or for example, chemotherapy, or radiation, or oxygen reagent. I mean, all those are insults. This guy can sense this insult, and in a good time, you know, when we are healthy, it can do everything that the opposite side of the fitness that I showed you before: survival, cell cycle arrest, apoptosis, and so on. So immediately, this is there. So you can even guess that if it is affected, all of this will not happen. All of these good things for controlling the stress will not happen. So I use this example to show you what people are doing now in the clinics. And during my talk, I will try to combine, or at least to push a little bit the border, how this research that I'm talking about reach the clinics. Okay, so sometimes maybe I'm pushing too much, but I'm thinking about it all the time. So there is a good catalog that is called COSMIC, which is the catalog of all somatic mutations. Somatic mutation means within the cancer sample. Okay, so it's happened in the sample of the cancer, not in the necessarily in the person. And what people are doing is what I like to call the frequentist view. What does it mean? You just count. You go to the catalog and say, okay, how many times I see a mutation in P53, for example. This is P53. The protein goes from here to here, about 400 amino acids. And you can see quite clearly that there are some hotspots in which thousands of reports, this is the number of reports in this COSMIC, 2,000 people have this amino acid changed to this amino acid in this position. So just so you know the terminology that I'm using. So this is amino acids, this is the number, this is the change. Okay, okay. So you can see, but if you look more carefully, you can see that it's all around. It's not that this is the only places. But the frequentist view is, of course, very valid because, indeed, in many cancers, this gene is affected. No question. I explained why. But it's very sensitive to many biases. For example, if you have a specific cancer type, I already showed you that likes more p53 than other, there are hotspots in the genome. Our genome are not, I said randomly, but they are not random. There are places which like to be mutated and places that are fully protected. So these hotspots will change, depends where the gene is, right, in the genome. And the gene length, this is very trivial. If the gene is very long, there will be a lot of mutation. So just counting is not enough. Even counting and correcting for length is not enough. Okay, so I'm just making this argument. And I want to illustrate it. I went to the NIH, I took all the cancer genes and sort them by the percentage of cases affected. So the most affected in all cancers. And of course, the top guy is p53, as expected, but quite surprisingly, the only 2 driver genes that are in this list are those that I marked with red star. Those are apparently with a lot of mutation, nothing to do with. For example, these genes, to those that likes those, this is the longest gene in our body, and so on. So you can see that there are a lot of biases here, and the question is, if we just do the frequentist, we are falling into this trap of telling something that is completely non-relevant. Okay.
Can I ask what this plot would look like for people who don't have a cancer diagnosis?
All of them have cancer, and now I'm asking.
What if you did this plot, but for people who don't have any cancer diagnosis?
No. Let me explain. I take 10,000 cancer samples, and I ask how many of those 10,000 have defect in TTN, and there were 32%, 33%.
And what percentage of people who don't have cancer have a defect in TTN?
Very few.
Very few.
Yeah.
So
yeah.
they're really, it's not just.
Yeah. It's not that NIH is doing so stupid things, but you know, sometimes they do. But what I'm saying is that the definition. This is why I talked about passenger and driver to separate those two. Okay. So the idea is we are looking for positive selection from the evolutionary point of view. What I mean by that, all genes that playing roles in cancer are expected to show sign of accelerated positive selection. Namely, they are changing faster than we expect by random. In a minute, I'll tell what is random. But what I mean by that, schematically, I don't know how many of you knows about genetic code or needs 1 minute to tell you. You need, okay, perfect. So I want really, it's important for me that everybody will follow everything, even. So this is a gene. And you have cases in which we have 3 terms. Synonymous means you change a DNA a mutation, so a single site, and you didn't change the amino acids. I'll explain later why, how come that it happens. So you change the code, but you didn't change anything. The nonsense is a really brutal change. Why? You change the 1 point, and then the protein is dead. Why? Because you put a stop codon. You mark this gene cannot be produced. So this is a bad guy. And missense is a change of 1 amino acid to another, which could be a broad range of impact, right? It's not. So for example, just visually, so if this is a neutral genes, just the genes that we just talked, and now you see that the synonymous are the same, but you have access of nonsense and missense, and you see them, so you say, okay, there is pushing here, a selection to push them to be more damaged than expected. So we want to understand, this is most of the genes, and this we try to understand. Okay, to do it in a more formal way, I would say that we want to separate the 2 scenarios. And for that, we develop Fabric. So now that you have all the background, I can go and explain what's Fabric. Fabric is a complementary system, instead of counting the frequentist view, it's a functional view. So I'll explain how we moved from counting to function. Okay. So the fabric, the idea, I think I said it, but I say it again. We want to identify genes that are more damaged than expected. We want to determine what is expected, because that's the most important part, to understand what's the reference. I mean, otherwise you can say, wow, I see a lot, but what does it mean? Maybe you expected a lot. So, and then we want to model the background while avoiding all those biases that I mentioned before. Okay, just for the people that are more into the genetic field, I would say that once we have this, we need some, I call it mapping machine, which is a machine, let's say, that transform a variant, a change, a mutation, as I said, to its impact on the function of the biology. Really to formalize better the word damaged. What does it mean, damage? Okay, so this is what we do. And to do this, we develop this machine learning. I'll be very fast on this because it's, let's say it's a machine, but we take the DNA, we take the variants, and we take everything that biology knows about. And biology knows a lot about protein, about genes, about coding genes. We combine them all with 11,000 numerical features, and we built a machine learning. We did it. We started with it even 10 years ago, and we did some engineering of features. I can talk offline about this, and we built this machine, and this machine was trained on ClinVar. ClinVar is a database, interesting also for you. It's a database of clinicians that once they have a mutation geneticist, they put it there. So it's a huge collection of everything that have been seen in the world. So we trained the machine over this, and the machine, you know, performance is pretty good, AUC is 90% and so on. So it's a good machine, but most important is that firm score, we provide a score for each and every change, trying to capture the likelihood of a gene to retain such and such percentage of its activity. So it's a probabilistic view. So if the gene is fully active, it's 100. If it's dead, it's 0. And everything in between is everything in between that the machine tells us how to infer. Okay, so are we good up to now? Because it's a bit a molecular crash course, but I want to make sure that we are following it. So one question which is legitimate, and everybody should ask himself, why did we do FIRM? I mean, there are so many of those variant-based inference. I just list some of them that are used in the clinic quite often. I mean, the reason being that we wanted only to say what happened to the biochemistry of the protein, what happened to the function of the protein, and we wanted the probabilistic intuition that I already mentioned, and we wanted it to be human-specific. That's a delicate issue, but I want to emphasize this. What I mean by that, a lot of the tools that I showed you before, just as a cloud, are using what we called MSA, multiple sequence alignment. So it's a matrix pile of all organisms in the world for these genes. So you can see that some site of the genes is completely conserved. So you can say, wow, this must be important because it's conserved from yeast to human. But we wanted something that will be human-specific. So we ignore all this information. So we had to build our own machine, let's say. Yeah, please.
So I'm sorry, it's been a while since genetics, but why do we need a probabilistic function for this? Isn't this deterministic? Like if I gave you the sequence of amino acids for a protein and then I would tell you that I mutated one of them, couldn't you tell me for sure whether or not this protein would function?
Not at all.
Not at all.
Actually, if you have it, you'll be very famous.
I'll be famous?
Yeah, so try to work on it. The reason I'm saying it, that let's say, again, I can talk a lot about it because it goes back to the early bioinformatics thinking. Naively, people thought that a change of amino acid, let's say from very positively charged to negatively charged, will kill the protein. Wrong. People thought that if you have something that will break the helix, from structural point of view, because now we have this alpha fold, we can do all structure. Wrong. So we have it's much more context dependent. You need to know which protein, what domain, what area, and then you can predict. Without it, it's not useless. It's very good, by the way. No, it's not useless. It's very good for evolutionary, long-term evolution. So like from yeast to human, you can use it quite nicely. But not when you're talking about mutation in human. It's the time scale is different. And I have a lot to say about it, but the answer is it's not trivial at all. Please.
Maybe I missed it, but for the biochemical damaging,
Say it loud, please.
The ground truth for the, so you're looking for the ones that have biochemical damaging score. That's your first bullet.
Yeah, I'll show you the example. It's not like 0-1, as you mentioned. I want to show that a specific site that happened, that I saw in the sample, in the cancer sample, I can provide it a score of 0.7 or 0.3.
But the ground truth for this is coming from ClinVar? Or?
Yeah, no. Okay. You have a point here. We trained on ClinVar,
Okay.
and ClinVar by itself is not biochemical. It's clinical. It's pathology. So this is really, we did this jump. But once we did it and the performance was good, we said, okay, now we are using it as a probabilistic machine to say what happened in each and every site that we see. And I think the demonstration is pretty clear on this. So what I wanted to mention, which is related a little bit to the question that have been here, we are very good in capturing the folding, the stability of the protein, the biochemical function, enzyme, not enzyme, and so on. Subcellular localization, we have Q where it's in the cell, the protein modification, the protein ligand interaction, and so on. But we are not good in some other aspects of the protein. And in some later development, we are also dealing with non-coding genes that I'm not talking now about, and regulatory elements like promoters. Promoters are very important to those that are not familiar with the words. Promoters are the switch point. It's like turning the light on. So maybe the protein is perfect, nothing happened, but the switch is gone. So it's as bad as the protein is not functioning, right? So for now, for this discussion, we ignore it. But I'm just mentioning it because it's always important to know what you know and what is still on your plate. Yeah, please.
Just to clarify, so for these genes, would it be correct to assume that we have good characterization of the functional sites of the structures?
Okay, yeah. So I can say that we are working on 20,000 genes, which is what we call the human proteome. So the stabilized human genome that we know fairly good. We have another study on the non-coding, which is 50,000 genes, which we know nothing about. This is even more crazier, but I won't tell you at all about it. So we picked, and especially for this discussion, I wanted to be on the safe side, always walk on a road that will take me further. So those 20,000 are mostly well characterized as a proteins.
And then just as a corollary, so when you have the score, so I'm not thinking as about
sure.
Right? So when I say, okay, 0.7 versus 0.75 or 0.76, what does that map to in terms of the changes
okay,
in
fantastic.
the functional site?
Can you keep this exact question for another 2 slides? Because this is the answer. Yeah, but it's a good question. Because I'm going exactly there. So before going to answer your specific question, which is a very good one, I want to say, okay, what we have. And that goes a little bit to your talk. We have 33 different tumor cancer with 11,000 samples open to the public, letter by letter by letter. Now, I talk with you a little bit, just as a side story. I was wondering how come that people, but this is on psychology aspect, they don't care to give their cancer because it's not part of them. I mean, this is my cancer. I mean, do whatever you can. So the cancer samples, there was no privacy, no issue. Take it and do whatever you. So early days, this 11,000, actually it's 14,000, but publicly it's not 11,000 because 3,000 didn't want to share. But this number we are using, and it's quite an amazing tool because, this is a comment, because for each sample, they have 7 layers of information, which is insane. RNA, DNA, clinic, everything. So for this specific talk, I'm only talking about DNA sequence because I want to stick with the mutation story. So what we did, we collected 3.2 somatic variant. Variant is mutation. I'm just changing the words, but it's the same. Across 10,000 that we had sample from 33 cancer type, and we filtered out only the subset, which is quite a lot, 2.2, that are within the coding regions, within the function themselves. No promoter, no leftover. Okay. So, because I think somebody noted saying, we want a brief on what are you talking about genetics, and so sorry for the geneticist in the crowd, but we know that the genome is ATCG, right? We know that the genome is super long. We have this number of nucleotide multiplied by 2 because we have 2 chromosomes for each. And each cell, we have this number of cells. I mean, big numbers. And we have hundreds of cell types, not talking even on cancer, that they have thousands of cell types. But just to say, and what important is this, actually, to those that do not know this number, it's shocking. Whenever I'm teaching, for 40 years, I'm always saying, wow, what's going on? Everything that I told you today is about this 1%. Because that's what we understand, for sure. So, or at least to a good extent. So I'm talking about this protein, or as we call them, coding genes. And the proteome, the words are not ATCG, but amino acids, right? As there are 20,000 genes with good knowledge, I mentioned. The genetic codes, which is really important, is reading this DNA. Let me just A, T, T, C, C, G, G, A, T, and so on. Let's say we read like everybody can see it. Yeah. We read like this, okay? The genetic code is read by triple. We call them codon. Okay? And there are 20. This, for example, this, by the way, by mistake, I did a stop codon. I didn't mean to. But anyway, this is proline. So you have an alphabet 1 to 1. I mean, once you have a 3, you know what amino acids will be there. It's no problem. So 3 nucleotide, 20 amino acid, 64 codon, because it's 4 letters multiplied by 3, and 3 of them are stop codon. Like by mistake I did here. Okay. So now, a nice way to look at it, now don't U replace T, so read it the same. If you read U and T, let's say it's the same for simplicity. So if you do C, C, and for example, if you have a sequence in the triplet, C, C, and then A, or for that matter, T, C, or G, all of them are proline. You see? So you have, this is the the synonyms. So you have quite a lot that can be replaced, the same without affecting the end product, the amino acids. Okay, so now that we passed this 101 class, we can go on. So let's go to your question very carefully. As I mentioned, we take this 10,000 sample from cancer, from TCGA, and we count how many cases of within P53, I'm just using it, then I'll do it for 20,000, but this is just example. And we found over 3,000 cases of mutation in TCGA. And then we looked, and 3% were synonymous, no change. 16% were nonsense, mainly stop codon. And 81% were missense, changing one amino acid to another. So what we do, we take the sequence. Now I'm running the sequence. This is amino acid. At number 36, there is a proline that was changed to serine. We ask our oracle firm, what do you say about it in this context, in this gene, with everything that you know? Say, oh, this is 0.63. We went to another one. This is a proline, but this is really likely harmful. It will kill the protein. As you mentioned, Noam, before, this is what we knew. This will be like this, and this is a change, but didn't change the amino acids. It was a leucine, remained a leucine, so it comes to be harmless 1. So once you do this, you have all the observed counting, so you have a distribution of numbers, because we count 3,000 of them. So the distribution provides us average, standard deviation, and all the properties of the distribution. So everything is good, but what can you say about 0.05? I mean, what does it mean? Nothing. So the trick is to try and to do a background model. This is where we compare. And the background model goes like this. I already mentioned ATCG. There are, not in principle, for sure, only 12 transitions. A can be G, can be moved by the mutation. A can change to T, A can change to C, but C can change to A, and so on. So there are 12 of those. Up to now, everything is clear. And now, this I already showed you. You read it C, C, U, C, 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 A, and this is proline, and so on. And here is a stop codon, and so on. Termination codon. So U or T, G, A, it's a termination codon. So now we go and we ask firm to tell us what the changes are and how you do it. We do it in the following way. We go to P53. Now, remember, this is a delicate point, but I like it, so I'm pushing it quite hard. The point is that P53 is sitting somewhere in the genome. So we want to overcome the fact that it's a hotspot, not hotspot. I mean, so what we do is the following. We take one of those changes, and then I'll multiply it by 12. A to G, for example. And every time in the sequence, this is the sequence, this is the amino acid of the P53. Every time we see A, we change to G. A, G, A, G. And each time, independent. And now we ask, what firm tell us about it? Okay, this change didn't change proline to proline. This lies into glue, and so on. So all those changes, this didn't change at all, this didn't change at all. So we count everything that happened, and then we ask firm, and we have distribution, again, of numbers of distribution, but this time we do it for all 12, right? Because we have 12 cases like this, so we repeat it to all changes. And one comment that is quite interesting, and again, there are a lot of explanation that is beyond this talk, but we did independent testing basically on all changes that could ever happen in any gene for any 12 changes. So we have a catalog of all background ever for each gene specifically. Okay? So we based on a lot of calculation, of course, and this 12 is, there is another correction point here. I talked about G to A, right? But look at A to G, the percentage in P53, completely different. Why? There are biology behind, but we had to compensate for this. So the calculation will be or weighted correctly by this table that is specific for each and every 20,000 gene. So now we have a background. I hope it helps explain, and we build up this fabric, which is a positive selected signal, and I think this summarizes everything that I said. The last part that I didn't mention is to take the observed distribution and then the theoretical distribution, compare them, and do exact statistics. Is it a p-value? Is it, I mean, what's the story here? And yeah, so this is what we do. Please.
Yeah, so on the previous slide, when you had the A to G and
Yeah.
those statistics, are those based on like empirical numbers over patients who may have cancer or may have the disease, or are they based on the background population, or like underlying biology? Or?
Yeah, it's the underlying biology, and it's pretty stable.
Okay, so it's not expected to be related to the disease process.
It's a good point, but no. Okay, so now you understand fabric from start to end, basically. And when we did it, I want to show you that fabric is also a discovery tool. Because when we did it, we found that 2/3 of what we found with no assumption. I mean, exactly as I showed you. 2/3 were known to be driver genes, which is a good sign, okay. I mean, we didn't put the word the driver there, but it came out to be so. But interestingly, 30% were new, were unknown. And we wanted to understand whether it's a noise, whether we are missing something. So so 410 were already reported, and this number was kind of new. We didn't know what to do with it. Just to show you that I mentioned Cosmic before. Cosmic report on 500 driver genes, and we have a very strong enrichment and a very good p-value. So we really enriched dramatically in some more restricted set of mutation signature, we have even higher enrichment. So we were pleased that we found what we looked for, but then I want to show you why was it important, because it's a completely complementary method. So maybe we just found what everybody has found, and that's good by itself. I'm not saying that it's bad, but whether we could do something interesting else. So let's take P53, which is in top of the list. If you look of how many mutations per nucleotide, it's really a lot, like 2.7. It's a lot. So everybody knows about this. And we have a very good score, and we have, but look at this guy, for example. This gene doesn't have mutation. So in the frequentist test, it's below the bar. But here we see that the number of mutations per nucleotide is low. However, the z-score that we are asking what is the difference between observed and expected is extremely high, extremely. So this is number 1 in our list in terms of z-score, and with great statistics and so on. So basically, we were able to come with a new, I would say, weighting that comes from function. So that's was nice, and but we were too curious to know what are those 180. So I called people. It was during Corona, during COVID. They didn't know what to do with me. You know, there is. But I wanted to get to the expert in those genes and say, do you know if this gene is expected to be driver? You know. So what happened? I said, okay, let's put all the 180 that are new in this list, which is the effect and the p-value. And we say, well, a lot of are in the noise, but let's take those that are strong effect and high confidence. What are they? So we took them, and just to show you that if you take those 5, only 5, out of 103, this is the name, this is the percentage of cases affected, 30%, again, a good number. And we could see this is, again, this is a survival curve. Namely, just to people that are less familiar with this curve, say, at this point, there are 100% of the people that live. Everyone that dies from this is reducing the population that they are all cancer patients. So the population of cancer patients that do not have a mutation in those 5, this is the blue one. And this is only the patient, the sample, that have mutation in any of these 5. So it's a completely disjoint group, and you can see that the survival is different. The median survival 17 months against 26 months. So, but we still don't know what they do, let's say, but still we have clinical implication that say, look at those. So this was, sorry?
Are they mutated in specific tumor types?
In this case, I did the pan cancer, and actually I also checked it to the in different, completely outside source of Memorial Sloan Kettering, which is a very strong but more advanced cancer. So yeah, we can talk about this. But so I think, as you mentioned, pan cancer. I mean, people or doctors are not treating pan cancer. They are treating either breast cancer or lung cancer or colon cancer. So we say everything that we did it for pan cancer. I mean, cancer is one term, which is, of course, not the right. So we did, this is what I showed you before. Now, each of them is one of the cancer type. So you can see that P53, as I mentioned in the very, very early of the talk, is shared with everything, right? But if we do, as you ask, about cancer specificity, we have a Z-score for the difference. And now you see that P53 is in the bottom of the list because it cannot specify specific cancer. So if you are looking for marker for a specific cancer, don't go for P53, but go to those guys that are somewhere in the middle of the list. Okay, so exactly the move from pan cancer to a specific cancer is very important. So like always, we put it to the public. So we have a website, everybody can use it. And the conclusion is really, we use minimal assumption. We were very conservative, so we prefer not to say something that we are not sure. It's fairly robust because we are working on 18,000 or 20,000 genes and not 2 million variant, and so on and so forth. And also, it's very good for rare mutation. So stuff that happen only once, so only it still can provide information. This is also quite nice. And Fabric is a human view proteome evolution, if I'm summarizing my subtitle. And to those that knows Nati, it was part of this project. And okay, and it benefits from more data. You have more data, you have more power, and so everything is good. So if I know that the time, if I have another, I'm good? Okay. So I want to move. So I put a lot of emphasis to make you think like we do. So now I feel that I can move a little bit faster to talk about population. And population, usually in the field of clinic, you use them as case and control. Those that do have the disease or the diagnosis, and those that do not. And the collection of data that we are using is quite enormous and quite, I'm working with it for 10 years now. It's 500,000 people from Britain, admitted to 23 hospitals and clinics, major clinics. They were collecting, this is what I like them. They collected the people, these 500,000 people, in a specific time. So they are not adding people, so they are just aging. So, and the records become updated. So you can do prediction for the first time because you haven't seen them later. So you can, this is really important point. And at that time, the age was 40 to 70. Now they are 85. I mean, so it's really, it's become better on time. That's my point. And what's there? Enormous information, medical, diagnosis, lab, procedure, self-report about interviewing, really long interviewing, lifestyle, nutrition, sleep, mental, X-ray, MRI, you name it. And the genetics become really, really detailed, as if there is no privacy issue here. I mean, really detailed. And what I write here is an all genome sequence. All genome sequence means the 3.2 10 to the 9th, each and every letter, for 500,000 people. This is a treasure for geneticists and so on. Just to show you that it's very detailed, this is 500,000 people saying what they like to eat in breakfast. What type of bread? Is it white or brown, with nuts, without nuts? So you have it. And you know, most of them, so now you know that the British likes whole grain and so on. But another question is how many sausages you eat in the morning. But as you can see, they didn't put 0 as an option even. So it's start with half and end in +5, and everybody, you see that at least 2 on the average and so on, and it's enormous type of, and the question is, do you like your beer in room temperature or cold, and you have it within the meal, before the meal, after the meal, cider, I mean, like crazy. And it's all there, and it's all in data field, very, very well organized. Just to say, because I was yesterday tired, I added another slide, which is what type of person are you, morning person or evening person on the sleep? And immediately you see that most of the people relate themselves as rather more morning type than evening type and so on. So you have a lot of information. By the way, some other data set that I don't think I have time to talk about it, it's called All of Us. It's a American style. They now added 35,000 people with watch, with the dynamic information on how many steps they went today, I mean, it's a new word in terms of, let's say, environmental studies, especially. Anyway, we wanted to develop, and now I'll do it very fast because you have all the background. So PWAS is a human population association studies, again, proteome-based, same way. We use machine learning. We use a rare and de novo variants, no problem. We use variant and not other type of genetic variation. And we use firm as a oracle for what happened, like we did before. And the goal is to find possible causal, but at least associated, link between genetic variant and human phenotype, like a disease or a human trait, like your blood pressure, for that matter, okay? So it doesn't have to be binary. So you do it, and why? This is the most important, because we want to understand biology. We want to understand mechanism, what's going on beyond this disease. So this is the only way to understand and how. We do a case control and also a little bit of a family that I don't think I'll have time. But what people are doing today, the same way that I mentioned these frequencies, that is the common knowledge, what are people doing today? GWAS. What's GWAS? Genome-wide association study. It's so simple that I can explain it in less than 10 seconds. What is this? You take the case, for example, those people with heart disease. You take the control, those people do not have heart disease. So the population, could be 500,000, is separate. And then you ask on a specific site on the genome, you have the DNA, what's written there in this specific site. In this case, it's C, 62%, and T, so on, and the control is like this. So is it statistically significant? End of story. This is what people are doing. So think about that the GWAS is very simple. This is why it's so powerful. We are talking about 10 million. I said common, but it's not common. Common and imputed. Common and what we know from other people. So it's called common imputed variant across the human population. The significant, to get a significant, you need a really high number. Why? Because you have so many hypotheses. 10,000, 1 million, I mean, a huge amount. So you have to pay a lot. And there is huge amount of false positive. To tell you the truth, it's a variant base, as if each variant is the story. But it's never the variant base. That's my point here. It's the minimal you can say it's a gene base, as I mentioned before. And the way that this data is presented, GWAS, it's called the Manhattan plot, because it's like you go, those are chromosome 5, 6, 7, 8, and each chromosome you go to all the millions of variants, and you say, this is the p-value that I got. And this is the line that below it, you cannot consider it real. So you have few peaks, like in Manhattan, and this is how people are using the Manhattan plot. However, 20 years after it came out, look at the number, 45,000 publications and so on. It's a huge field. I would say that it came out a little bit disappointing. And why? Because there was no biological meaning. Because it was poor utility and very non-applicable. And third, there is a poor mapping of variant to the function. I mean, okay, you see a variant here, what does it mean? Does it affect this gene or the other gene or not at all? So the interpretation is hard. So we decided to do exactly what we did before. Instead of jumping from the variant to the phenotype, to do it through the protein that we know so well. So we did basically this trick. And the trick, I'll have another addition. You know, we are in a strong genetic class now. Usually, when you do GWAS, you have an assumption, a naive one, but it's working. If you do not have a mutation, you call it 0. If you have one mutation, you call it a variant, because we are talking about population. The word mutation is not relevant because it's what in us, basically. You say 1, and if you have in 2 places a change, you say it's 2. So it's an additive view. But we wanted to say that if we Mendel, when he described gene inheritance, the concept of inheritance, the concept is about dominant and recessive. What does dominant? Dominant say that it's enough that you have one effect, and the full function is up or down, for that matter. What's recessive? You need to have at least 2 changes in order to get to the mutation. So we wanted to add this layer, and we say we'll do recessive. Namely, we would like to capture 2 mutations in 2 alleles at different position within the coding gene. We are like this. We want to say that this is as bad as, because this was affected, and if firms say that it's really badly affected, this gene is gone. Now, this cannot be viewed in GWAS at all, this recessive aspect. So now I'm coming to kind of conclusion. So we take each and every person of the 5,000, 500,000 people. We go to 20,000 genes. We ask the firm, what is the score of firm per gene per person? And now we do 2 models, whether it's recessive or whether it's dominant, as I explained before. So now we can take the population of cases, let's say cancer, those with no cancer, and do the distribution of the recessive and dominant, and just do exact statistic, exactly as I did before. So when we did this, I'll show you what we find, but by that I kind of wrap up. Remember that as of now, I talked about fabric in the first part, and fabric is just, one can say, bad luck. You know, it's cancer, bad luck. Maybe the two-hit hypothesis, maybe mosaic from embryo. I mean, but so this is what we did for fabric. But now we are talking about population, what we call germline. You were born with it. This is why it's a predisposition. So you have the tendency for a cancer in your genes. That's what we like to find. So through population and through families. And because I talked about population, I should say this graph is quite interesting because it says the different type of cancers have different percentage of what is expected to be genetics. I mean, the source. So for example, ovarian is very genetics, 20%, it's a lot. And some others are not so much genetics, but there is a component of genetics in many of those cancers. And if I have to summarize 40 years of work on predisposition, basically it ended up with a very simple tables like this. So for example, for breast cancer, you have BRCA and some others. For this, you have this. For colorectal cancer, you have APC. And it's simple. But we ask ourselves whether we miss something. Maybe. And what we did very simple, what I told you. We took the 500 people. We filtered them out to keep the Caucasian only. Again, for reasons that are important because different ethnic groups have different set of genes. So, not set of genes, set of mutation and predisposition. So, we used the cancer registry. I mean, it did have a cancer and what type and so on. And we removed family member. If there is a big family and so on, we took only one representative, not to bias our sample. And then in this set, there were 20% that have cancer. That's a lot. So now you can have case control in a fairly good, we did it and we found 137 unique loci. Places where, now remember the list was of size of 50 after 50 years of work. We just came with a different perspective. And we learned a lot from this analysis. I don't have time to go through, but one interesting finding that I think a lot of clinicians will know it, but I'm saying it because it come out from our data without knowing, is that many cancer, I'll say it differently, because in somatic mutation, like in cancer samples, there were P53 and people and genes that were united to all cancers. Here it was the opposite. Each cancer type has its own gene, and that's it. So it's a specific predisposition to specific cancer. It's not general, which is quite important and interesting. This is what we found as new. I won't have time to deal with it. But just to show you that if you would do a PWAS, you couldn't get it because it's below any p-value. If we did the PWAS, we call it PWAS because it's a proteome WAS. We got a very nice statistic. But it's not the dominant, it's the recessive story. This is why it could not be even found. You see, so we found, because we found the switch of the recessive, and actually, just to wrap up, we say, this is what PWAS found, this is what PWAS, and this is what both agreed on finding. So there were quite a few that there were no evidence whatsoever. There were, in all our list, 2/3 were recessive that we didn't even think about, that there is a recessive aspect to cancer predisposition. But that was an important discovery. And I would say, I think I'll stop here. I'll just say maybe one slide for the clinics. Well, how this may be nice or not nice, but how this very theoretical aspect can get to the clinics at all, if any. So first of all, we provide lists with a level of confidence that we believe, which is by itself, it's okay. Maybe you can put it in some additional data or panels in the clinics. By the way, those comes from panels. So there are already tested for in the clinics. So it's not that we made up something that is not in the clinic. And what we did something that is a little bit, maybe the word is not like PRS for the more professional people here. We rank all the population from the most severe one by firm to the least one. And now we ask, can we split the population so that we can see the two sides of the population, what is the risk of this side and of this side? And this is what you see. You can see that in this case, this is a very rare, I wanted to give an example of a very rare disease, that the prevalence in the population is 0.1%. So there are only 500 people with this in the, but if you divide it to 2, you can see that those guys have 15 times more risk than the rest of the population. So you can really, each and every gene can split and say, is it informative to the clinician? And you can say, unless it's 100 times, I don't want to look at it. So you have cases of 100 times. So this is kind of nice to the clinic. And last minute? Oh, I'm good? 1 minute? I can stand here for another 2 hours. You don't want it. So, last point is, we talked about cancer, and I mentioned that I took it as a case study. However, most diseases are not cancer. Most diseases are common diseases, like here. This is a percentage in the population, aged population, above 65 and so on. Actually, the 10 common chronic disease, 80% above age 65, that I'm deep there, have at least one chronic disease. And 68 have at least 2. So we are talking about the real burden on the economy or health care in general. So what we did, we built up PWAS, and now we did it for 500 diseases. The same way that I showed you, and we provide it as a web tool in which people can go through the gene of interest, through the disease of interest, through the association of interest, and so on, and get some result. And we did it also for male and female. And I won't show you the data because it's too long, but I can show you cases in which, without separating female and male, and do the analysis separately, you won't get the right signature. And even in diseases that are not supposed to be gender related or sex dependent, such as hypertension, we see that hypertension is by far the signature in women is stronger, 10 times stronger than male. And we also were able to show immune component in the women genes. So we got into the somewhere closer to the mechanism just by doing this as not using sex as a covariate, but rather than as two groups, as if it's two populations. And that also helped a lot. And I think I'll stop here. I'll just say, just to say that this, I wanted to show you that this. I talked about biobank, but there are a lot of biobanks going on, and the comparing between the biobanks are super important because there are different population, different ethnic group, and so on, and that's what basically we're doing. And whoever wants to read more can come, and I'll tell you. And thanks to all those great people that did the work. I'm done. And sorry it took so long. I didn't mean to, but you were so nice audience.
Perhaps just...
we can do it later.
Yeah. For lunch, we'll have this further discussion over lunch.
Yeah. I'm here for the week, so... yeah, thank you.
Thank you.
Yeah. One thing, there is lockers just kind of at the end of this whole